All right, everybody, welcome. I know I've been talking about doing this for a while, but I like to do everything live, and that means a lot of work had to be done so that I could do this live. Welcome, I'm Matt Dillahunty from Epic Loot Exotics. We are exotic reptile breeders of crested geckos, corn snakes, hognose snakes, womas, African house snakes, but primarily ball pythons. And frequently I've been asked to kind of describe why we do this, what's behind all this, and I'm fascinated with the science and the genetics behind it as well. And so I thought I'd make this easy and start by showing you a picture. These are all available at morphmarket.com. Credits to the respective photographers there. This is a picture of one of many examples of a normal or wild type or classic ball python. This is what's existed for millions of years, or what has been the result of millions of years. This is what you'd find in the wild if you were just to go out and find one. And so over time, the genetics of these snakes change based on pressures in the environment. And we resulted with this as what is the normal wild type. Now, this is not the only example, because here's an albino, and this particular morph, which morph's just the label we use to describe visual changes in snakes. This was a trait that was proven out in 1992 by Bob Clark. And it's the reason I wanted to get into breeding ball pythons to begin with. And Bob Clark also helped pioneer a number of other traits. This is an albino or amelanistic corn snake, so not a ball python. This was first produced in captivity. It says here in 1959, but my understanding is that it's 1961. The first wild albino corn snake was found in 1953. In 1959, it was bred to its own offspring in order to produce the first captive bred albinos in the corn snake family in around 1961. In 1986, you have this, which was also produced by Bob Clark, which is an albino Burmese python. These were the first of the larger and pet trade pythons that were available in an albino variety, also amelanistic. And that eventually led to the one I already showed in 1992, this particular, or not this exact snake, but this particular morph of albino. Now, as you begin to look at the complexity of the variety of snakes that are available, what you can get to is this. This is the current list of available morphs listed at morphmarket.com. Now, it's not exhaustive. There are always people who are working on new morphs, new color and pattern varieties and things like that. And some of these are duplicated. There are a number of different genetic changes that have been independently discovered and therefore were given different names by the individuals who found them. If you find a morph, you essentially get to be the one who names it. And so sometimes you end up with several different morphs being independently discovered and named by different people. So while it appears that there are about 300 or so, 250 to 300 morphs in ball pythons, it's actually slightly less than that from what we've been able to actually verify so far. Now, when we're talking about ball python morphology, we're typically talking about color and pattern morphs. And we can also look at the snakes in different regions. There's the head, the neck, the body, the tail, the belly, the eyes, and there's also additional morphs like scaleless. I've got pictures of scaleless head patterned ball pythons that in the super form produce a totally scaleless snake. And this trait's been produced in other snakes as well, Texas rat snakes, corn snakes, things like that. The difference being that those snakes retain their ventral scales, the scales, the wide flat scales on the bottom of a corn snake. They just lose the scales on the top, whereas the ball pythons not only lose all of their scales, including their ventral scales, but they even lose their heat pits in many cases as well. Now, the prices of these snakes, which we're not going to go into today, are dependent on how desirable they are and how difficult they are to produce, like the prices for anything else if we're talking about this as a pet trade. I'm going to set aside the price stuff because I'm in this for the fun and the genetics and things like that right now. So what's going to follow here is not going to be a proper, thorough introduction to genetics or breeding, but we will define some terms and concepts, and this should be a good enough introduction to give you a little feel for what's going on genetically and what the breeders are attempting to do. So the first thing to talk about is the difference between genotype and phenotype. And this is really easy. I'm going to make all this as easy and entry level as possible. Genotype is whatever information is in the genes, and phenotype is whatever information you see displayed in the creature. And so you could have a gene that doesn't appear to change the snake at all, and it would be in the genotype, but it's not present in the phenotype. The phenotype's all about what we tend to see, the things I listed before, like color and pattern changes, or reduced scaling, or elimination of scaling, all sorts of things. We're going to be looking at simple Mendelian genetics. We are not today going to look at polygenic traits or sex-linked traits or parthenogenic processes where a snake can give birth to an offspring, or a reptile or anybody could give birth to an offspring without having a partner. And so generally we're talking about a creature that has a mother and a father and that gets half of its DNA from each of them. And so if you were to imagine a strand of DNA, there is some section of some length that includes a gene. That section, that region, is called a locus. It is the location of a gene. And a gene is just that region of DNA that resides at a particular locus and is viewed as an instruction to do something. An allele is just a different version of a gene at a location. And coincidentally, this was not planned. I just happened to find this on my desk. I have here 2 copies of the Dillahunty's family recipe book. My mom put this together for me, and I lost it, and then I found it after she gave me a second one. But if you were to look at page 1 in both of these, they are not the same. So each of these page 1s is a page of instructions, and they've been hand copied by my mom, and they are not the same. There are slight changes, and this one actually has a whole second recipe. And so if you were to talk about this, what you would say is this particular sheet of paper is a locus. It is the location where we store the recipe for cheese ball. And the version of cheese ball that's on this page is different from the version of cheese ball that is on the other page. That's because copying errors happen. My mom made a change. Instead of mixed together, she wrote mixed TOG with a colon. On this one, she's got cheese ball underlined. On this one, she doesn't. The formatting is slightly different, but the recipe is essentially the same. It's how to make a cheese ball. And so there's copying errors. Now, imagine that we were a little more complicated and we needed everything on both sides of this sheet of paper to tell us how to make a cheese ball. And my mom copied one of the 2 sides of her page, and my dad copied one of the 2 sides of his page onto here. We'll get rid of the cheese ball instructions and we'll go to something much simpler. Let me pull up my example right here. This is a Punnett square, and it's how we go through and analyze what's going on with combining genetics. So you've got a locus, which is essentially that page. that contains the allele, which is some version of a gene that tells you how to do something. And each locus gets copied one half or one side of the page from the mother or the father, excluding parthenogenesis. Let's imagine that instead of all of those words, we either have red, just the word red, on either side, or the word yellow on either side. If the page has red on both sides, the two sides match, and it's known as homozygous. It just means it's the same on both sides. If it says yellow on both sides, it's homozygous for yellow. If it says red on one side and yellow on the other, then it's heterozygous for red and heterozygous for yellow. Remember, we're still talking about DNA strands. We're just using the pages as an analogy. And so at every location in the genome, in diploid creatures, you will have two genes, and they either match, in which case they're homozygous, or they're different, in which case they're heterozygous. If we'd imagine, for example, that on page 15 and 16, which is one sheet of paper, we have that red and yellow, and 15 and 16 is where we get the instructions for changing the snake from the default color. So if we assume that the default color we're just going to paint every chromatophore is just going to be black for the entire snake, unless the instructions here say either red or yellow. So if you look at this, if we say that the mom, for example, and the dad both had red on both sides of their sheets, then no matter which side they pick from and copy, every one of the babies is going to end up with two copies of red. If, for example, the father has yellow on both sides, then every single one of the babies is going to be one copy of red and one copy of yellow. They're going to be heterozygous for red and heterozygous for yellow. And the question then becomes, what does that do to the phenotype? If this is what's in the genes, what is this doing to what we see in the snakes? This is where we get into the issue of dominance. And dominance is not a value judgment. It is not a universal. It's not like red is dominant. It is a comparison of the relative dominance of the two genes at a location and whether or not they are dominant over, for example, the wild type, the default type, something at another location. We're going to set that part aside for a little bit. But if a gene is dominant, like we can make the red gene here the dominant gene, and you won't see any change in the graphic right now because all of these have red. And if we made the yellow gene dominant, you won't see any change in the graphic right there because all of them have yellow. But if we were to make the father red and yellow, now we have 4 potential offspring. They could get 2 copies of the red. They could get 2 copies of the red. Or they could get 1 copy of the red and 1 copy of the yellow. Or 1 copy of the red and 1 copy of the yellow. When you're in a position like this, if red is the dominant gene, then you will get a red display. Basically, the graphic here is using red, not because the R is supposed to represent red. It's just that it's using red to show the dominance. So if red is dominant, then all of the babies will appear to be red. If yellow is dominant, then half of the babies will appear to be red and half of the babies will appear to be yellow. And if neither of them is dominant, if in fact they're recessive and it requires 2 traits, or a homozygous red, then only the ones that are both red will show any sort of phenotypical display with color. So in a dominant, if one allele is dominant over another, then that's the one that will be displayed. If they are codominant, for example, if we're going to stick with red and yellow, if red is dominant, then the snake will appear red. If yellow is dominant, then the snake will appear yellow. If they are codominant, then the snake will appear red in some areas and yellow in other areas. Now, remember, this is an overly simplified view where we just have either a red snake or a yellow snake. There's a lot more complexity that goes in once we start looking at the genes and what they're going to do to different patterns. And I'll show you some of that in just a minute. If they are incomplete dominants, if red and yellow are incomplete dominant, then you'll get basically an orange snake. It's more like a blending of the red and the yellow. So you have either red is dominant over yellow, yellow is dominant over red. They're codominant, which means you get a part red, part yellow snake, or they are incomplete dominant, which means you get a red and yellow snake in the sense of orange. You can do that as an easy example because we're just talking about simple colors. It gets a little more complex and it's much more difficult to tell what's in a snake at different times. I'm going to show you, for example, this is one of the incomplete dominant morphs in ball pythons. It's fairly popular. It's called Orange Dream. Just for clarity, I'm going to go back and forth between Orange Dream and the normal. When you look at this normal, there's a lot of pattern, color, variety in the normals. Like I'll just scroll through a few. These are all normals. And yes, some of it's going to be differences in camera, but essentially you've got very dark browns, lighter browns, some yellows and golds, a little bit of white lining around some of the patterns. And then what you have in the pattern are a mix of what's been called keyholes or alien heads, and the number of holes that are in them varies. These are all just classic wild type snakes. And you can see on this one, there's some chaining of some of the heads together here, and some of them don't have eyes and things like this. These are all examples of normal. And when you take a look at something like Orange Dream, you can see this, I hope, that it's a little crisper, that it is slightly oranger. I have other examples of this snake. It just looks slightly oranger. Now, what ends up happening is that somebody will spot a particular snake that looks different from everything else they've seen. And they'll then breed it to try to prove out whether or not it's a genetically reproducible trait and figure out whether or not it is a dominant, incomplete dominant, codominant trait or a recessive trait, which means a recessive trait, you have to have, remember, a homozygous gene for that. You have to get genes from both parents that produce that. We go through a few other incomplete dominant traits to start with. So that's Orange Dream. This one is called Banana. It is a single incomplete dominant gene. You can see how dramatically it changes the color from the normal. It becomes a much lighter banana color. And amusingly, the banana gene tends to get these spots just like bananas do, which I always found extra funny. You can see that in addition to the color change on the back, there are also possibilities of color changes on the belly and in other areas. This is another example of banana. By the way, banana is also called coral glow or white snake. White smoke. And so that's banana. This is bamboo. I don't even know if I need to switch back. I can show you the banana again. So there's banana, there's bamboo. And you can tell these are the same species of snakes. They have similar shape, heads. There's lots of different ways to spot the phenotype changes. Some of them are about color and pattern. Some of them may result in lighter heads or head stamps, change the eye stripes along the side. For example, this next snake is an acid, and acid makes the alien heads much more drippy, as if someone were tripping and tried to look at a ball python here. There's also a little bit of a head stamp here. And then now we're looking at recessive traits. Piebald is one recessive trait where you need to get a copy of the gene from both parents. So a piebald bred to a normal will never produce a piebald. It will pass on its genes, and every one of the babies will be heterozygous for piebald, which means they will have one copy of that gene, but none of them will display piebaldism. You have to get it from both parents. This is why, if you remember the chart that we drew up, each of the babies that you produce, you would need to breed that to something that is homozygous or get lucky in a heterozygous transfer of a gene in order to add additional genes in there. So right now we're just looking at single genes. In addition to piebald being a recessive trait, you also have lavender albino, regular albino, T-negative albino, T-positive albino, lavender albino. These are all recessive traits. So you have to have the gene from both parents. But yeah, this is lavender albino, which is distinct from a regular albino, which is a much lighter yellow. And one of the things about regular albino or T-negative albino ball pythons is that they tend to fade out to be kind of whitish yellow as they get older, whereas the lavender albinos tend to hold their color a little better. As babies, the areas in between are this bright white. So you've got all of these potential snakes. And here, for example, I pulled this up just so I could show you a picture. This is a pastel scaleless piebald ball python. You can see from the picture here that it does not possess scales along its back. It also doesn't possess scales on its belly. And if you could actually get a good look at the face here, it does not have the holes where the heat pits normally are on the front of that snake. So pastel is an incomplete dominant trait. You can see it listed here in blue. Super scaleless head is the super form of an incomplete dominant trait. And then piebald is a recessive trait, which means you had to have it from both parents. And also, in order to get a super scaleless, you'd have to get that from both parents as well. If you have an incomplete dominant trait and you have one copy of it and you're heterozygous for that, it will display whatever its instructions are. And whether or not you see it, or to the extent you see it, it depends on what other genes are there and whether or not they're more dominant over this gene. If you have two copies of an incomplete dominant trait, you get what's called a superform. In this case, you're seeing two copies of the scaleless head gene result in a super scaleless head, which is a snake without any scales at all. I mentioned before that the normal wild types have a lot of variety present just looking around, and it's often very difficult to tell what genes are in a snake. Is this a normal? Is this a normal? As a matter of fact, I'm in a couple of ball python identification groups on Facebook, although I've left a handful of them. And one of the most common things that happen is somebody posts a picture of their perfectly normal ball python, and then they're like, this doesn't seem like it's normal to me. Could you tell me what else might be in this? And so people will look at it and it's like, maybe there's something else. In many cases, you can't tell until you breed them. Even for incomplete dominant traits, they're often hard to distinguish between from the normal variety, and you often need to get superforms. I have, for example, this right here. This is an incomplete dominant form called lesser, and we know that this snake possesses, at the location for where lesser always resides, this snake has one copy of the gene that we call lesser. It is heterozygous for lesser, and that displays with this. It's also called butter. Butter and lesser are the exact same gene, just discovered independently. And so you can see this kind of butterish color rising up there. I'll pull the normal back up again so you can see the difference between the normal and the lesser. So that's one copy. If you breed this snake to another lesser, you will get normal snakes, you will get snakes that are heterozygous for lesser, and you will, 25% of them, get snakes that are homozygous for lesser or are super lessers. And a super lesser is also called a blue-eyed leucistic. When you get 2 copies of, this one's also het clown, but that's not doing anything visually. It's just carrying a single copy of a recessive trait. But when you get 2 copies of lesser, you get this gorgeous, blue-eyed, almost completely white snake. The lesser gene that reduces the pigmentation to this with 1 copy will reduce it fully, and you get just a gorgeous blue-eyed Lucy. These are, we will be producing some blue-eyed Lucies at some point. There are a number of other snakes, including black-eyed leucistics and other complexes that we're going to get into in part 2. Today, I just wanted to go over just the basics of genetics, make it so that people aren't afraid of it. Basically, you've got genes, and versions of those genes that are slightly different are alleles. These are copying translation errors. These alleles reside on a strand of DNA at a particular location or locus, which you can think of as analogous to the page in there. In diploid species like this, where they get their genes from their mother and their father, each side of that locus is written to by the parent, and the parents are going to give one side of whatever their locus is there. Which means if I come over here to the calculator, right here, I know you guys can't see it. I've made it as big as I can. What we've got is a normal ball python that we bred to a ball python that has both banana and black pastel. These are 2 incomplete dominant genes. And because of that, we have, for each of the babies, there's a 25% chance that they will have none of those traits. Well, they'll still be normal because the normal one just wins out. But they won't have banana or black pastel. 25% of them will show up as black pastel. 25% of them will show up as banana. And another 25% of them, odds are, will show up as banana black pastel. Now, these are the odds. That doesn't mean if you have 4 babies, you're getting one of each. You could get 4 normals, in which case the odds were just kind of against you. You could also get 4 banana black pastels. As a matter of fact, we recently bred a banana black pastel mahogany to a banana that was het for piebald and het for lavender albino. What we got were 4 snakes. One of them is just a banana, which may or may not be het for piebald and lavender. We haven't tested. We got 1 snake that was a super banana mahogany, and we got 2 snakes that were banana black pastel. And I'll just click this and pull up a banana black pastel. Wow. There's a bunch of banana black pastels here that have additional genes. So if I go into this and set the max genes to 2, now we have, there we go. That's a banana black pastel. And so you can see that is roughly what we're looking at. I've got 2 of them down the hall. I'm not going to go get them today, but the black pastel makes this area that isn't yellow look a little more lavender, and it takes the alien heads and kind of opens them up a little bit, adds some pixelation in there. That's a standard trait of black pastel. You can tell the difference between that and, for example, this regular banana that is a little older, and so it's yellowed out a little bit. So the point of today was to go through and define some of the terms real quick. So you know the difference between genotype, that's what's in the genes. Phenotype, that's what you see. That is what is visible or that you can detect in some way. And in normal diploid creatures under Mendelian genetics, You have a location, a locus, that has a gene, or better yet, an allele. I'll get to the questions here in just a second. Has an allele, which is just a version of a gene. And then you either have those two alleles match, in which case it's homozygous, or they're different, in which case it's heterozygous. After that, what displays in the snake depends on, do you have a heterozygous gene? And is it dominant or recessive? For example, here's an incomplete dominant heterozygous for lesser. Here's an incomplete dominant homozygous for lesser or super lesser, and it dramatically changes the snakes. A normal here, it could be het for piebald or lavender albino or any recessive trait, and you'd never know it because it doesn't do anything visual in the snake until you get 2 copies of the gene, at which point then you get things like piebald and lavender albino. I'm going to answer some questions real quick, and I'll be working on part 2 of this, which I like doing it live. I get to answer questions and everything else. But part 2 is going to be dealing with the superstripe complex. So we're going to deal with allelic, so both superforms, and then allelic combinations, which are often described as acting like super. And that's because, let me show you these here. This is yellow belly. It's an incomplete dominant gene that's part of the superstripe complex. And the easiest way to spot it is this kind of light flaming that is coming up from the belly between the alien heads or keystones that's there. That is a gene in its super form. So if you were to find a super yellow belly, you get really close to a white snake, but it's not a blue-eyed Lucy and it's not a black-eyed Lucy. It's called an ivory, and it looks a little bit different. At this same locus, on this same piece of paper, you can either have 1 or 2 copies of the yellow belly gene, 1 or 2 copies of the spark gene, 1 or 2 copies of the spectre gene, 1 or 2 copies of the asphalt gene, and 1 or 2 copies of the gravel gene. And what you'll find when you're shopping for this is often you can't tell the difference between yellow belly and gravel and asphalt. They're just too similar. And when you breed these together, if you only got 1 of the 2 genes, you can tell that you got 1 of them, but you don't know which one it is. So they'll have to list them as gravel and asphalt. Now, go back to my full screen page, because what's coming up in part 2 is we're going to take a look at the superstripe complex, specifically some research papers that were done on both of them, both to describe how they use coloration in zebrafish as an example to describe the coloration in ball pythons. So you have your wild type form, then you have your altered patterning, which it starts at the dorsal and comes down less, and then you have striped, and then you have this white snake. And this is because yellow belly is the most severe phenotype in this zygote. Asphalt is the least severe. And so if you have 2 copies of yellow belly, you get what this ivory snake. 2 copies of spark, you get that. 2 copies of spectre, you get that. But if you combine yellow belly and spark, you get this snake. Yellow belly and spectre, you get this snake. You get a variety. And this is called the superstripe complex. So when you put 2 genes together, you get something that looks like a superform, in which case it's doing more than you would expect, and it's surprising. Not every allelic combination does anything fancy pants. And so that's going to be part 2. I'm going to try and get to some questions real quick, and then we're going to end this stream, and I'm going to go get some other work done. Thanks, everybody, for hanging out. So let's see. I don't know much about snakes. What does the word produce mean in this case? Well, that was a little while ago, so I don't know what I said, but snakes breed and they produce offspring. And now I'm getting booed for doing a short stream. Bummer. Snakes breed, they produce offspring. But when we talk about what do genes produce, when we talk about the phenotype, we're saying if you have this copy, 1 copy of this yellow belly gene, it will produce this visual effect in the snake. I think that's what I meant by produce at the time that you asked it, but that was a while ago, so I don't know. We have, let's see, as locus or locus pocus. Is it always 25, 50, 100%, or can you have like 3.76%? So I was just doing simple calculations with a single allele. And what I can do is show you this. So this is a Punnett square calculator, and you can change the number of traits. So that's what it looks like with 2 traits. That's what it looks like with 3 traits. And you can define what these traits are and do a cross match. Similarly, when I go to the ball python genetic calculator, let's say from the snakes in our collection over there, I have one that's a banana, black pastel, mahogany, and I'm going to breed that to a GHI inchy piebald. Now, every gene that I just typed in except for piebald is an incomplete dominant, which means it can be passed on from either, and you can see it. If the piebald, because it's a visual piebald, the parent is homozygous for piebald, means every one of these babies is going to be het for piebald. Guaranteed. That's how we pass stuff on. But you only have a 1 in 32 chance of hitting the all gene snake, which is a GHI banana black pastel inchy mahogany het piebald, because there are 32 possibilities. Now, some of these are essentially, I mean, you're not going to be able to tell them apart. You're not going to be able to see the het piebald in any of them. But your odds are based on how many different genes there are in the parents. And that's why the snakes get ridiculously expensive. I'll show you that real quick. We're just with ball pythons, we're going to sort by lowest priced first. And you can see that ignoring shipping right now at Morph Market, you can get, well, an inchy even. So let me pull up this inchy. Inchy is a location incomplete dominant where it changes the pattern so that there's more banding over the top. It's a little hard to describe, but it is at least distinct. You can get an inchy or a normal for $20. Shipping is going to cost you $70 to $100 or $40 to $80 if you were to do that. If you want to go highest price ball pythons right now, I'm going to guess it's about $30,000. Yeah, $30,000 for a pink snake. Then this snake, for example, this Sunset Hypo Mojave is $27,500. And so this is 1 incomplete dominant gene, Mojave, and 2 recessive traits, Hypo and Sunset. That's right now is the listing for that price. Let's see. How come one parent has only one of the 4 options? Isn't it supposed to be 2? I'm going to guess maybe we already answered that question. The parents have whatever genes the parents have, and each of them copies half one side of their gene to the offspring. So you get a combination of the parents. If you had, for example, if we went back to that calculator, which should be here, you could have a normal snake and then pair that to something with 12 genes, although it's getting kind of ridiculous at that point. But we can just do inchy. What the hell? GHI, lesser. So now you have one parent that has nothing beyond the normal and one parent that has 5 genes, and you get 16 different possible outcomes. You get inchy or black pastel or inchy lesser. Basically, it's every possible combination, which is why we do it in Punnett squares fairly simply. Let's see. Hi, Matt. This is Hannah. Hi, Hannah. Nice overview. Thanks. You're welcome. Thanks for hanging out. Makes sense now. If one parent's normal, they all have normal plus the other trait. So here we go. Here's one where you have a normal and you're pairing it to a snake that has banana and black pastel and inchy and GHI and lesser. And you can get, well, I shouldn't have put inchy in there because that ends up just being dominant. So normal combined with this, you can get a lesser or a normal or a black pastel or a banana or a GHI or black pastel lesser, banana black pastel, GHI lesser. You can get, any comp, any pairing of 2, any pairing of 3. And then there's the 4-gene snake, which is the GHI banana black pastel lesser. Boom. Are the tools needed to examine the gene common? No. How can you tell you're not getting scammed when acquiring a certain type, especially when the details are sometimes hard to discern? Good question. So there's a company now called Rare Genetics Incorporated. They are able to do genetic DNA shed testing. So you can take a shed from a snake, and right now they've only got a handful of genes that they're able to actually test for, but they can tell you for those genes whether or not your snake doesn't have the gene, has the gene in a heterozygous state, which is 1 copy, or has the gene in a homozygous state, which is 2 copies. However, we only have that testing for a handful of the potential genes right now. So the tools to test the gene barely exist. There's a company you can send your shed to. You can't test for genes at home. The only way to do it is to breed them and prove them out. And so the best way to do it, I wish I had a picture here for this. I'm getting ready to take some pictures because we have a clutch of 7 babies. I'll do another live stream with these 7 babies where we identify them. Probably not later today, but we'll try and do it soon because they're all in there eating right now. We know the genes of the parent because they're displayed. So the parent, like imagine you have this snake that is a lesser. You can have, let me go to the gene list, which is right here. So, nope. Morph market, ball pythons, traits. And so let's say we have lesser. We just click on lesser. Where is it? Right here. There are 2,180 snakes with lesser in them. If I sort them by lowest price, what you should see first is just a lesser. And then let me find a lesser with something else. Here's a pastel lesser. So that's our lesser. Get this embiggened so you can see it. And that is a pastel lesser. You can start to see, as you add more genes in there, what else happens. And the way you identify them visually is by comparing them. We know that, actually I don't really like that particular lesser or that particular pastel, but we know that this snake makes it lighter, this makes it darker, or this gene makes it, this allele makes it lighter, this allele makes it darker, this allele changes the pattern, this one changes the width of the eye stripes. There are plenty of little visual cues to show what's going on. And so I think probably this might be the best example here. This banana and this banana black pastel. When you look at this, a regular banana, and it'd be so much better if I had I have bananas and banana babies down the hall. But to answer your question, Edie, there's no guarantee that you're not being scammed, because when people list the snakes here, they're listing their visual assessment based on what they know they bred and what else they saw in a litter. And so we have a clutch of 7 babies down the hall. They came from the same parents. Arden says, you want me to grab a snake? Oh, you're done with your workout. Sure. Grab, grab 2 of the bananas to start with, and then we'll grab 2 from the other clutch. Grab the pastel gravel and the super pastel gravel. That'll work. All right, so we'll get some snakes in here. Hopefully this works out. But we have, you tell by looking and spotting what's going on, but it's possible, especially if you end up with a white snake, for example, like a super yellow belly and ivory, you could have 5 other genes in there, but because the super yellow belly washes it all out, you won't know. And so what tends to happen is people will list, hey, I'm going to give you, or I'm going to list it as super yellow belly, and then I'm going to say possible inchy, possible black pastel, possible all these other things. Let me go here to the big screen.
You said these ones, right?
Yeah, we'll start there. So these 2 came from the same clutch. Oh, you haven't finished shedding. You're still a little messy here. This, we did by comparison. You can see the blushing coming up the side. It's hiding its head, but you can see, wow, how white and washed out that head is.
I think the lights are bright. Maybe not.
This is a super pastel gravel lemonback, maybe inchy. I'm not completely sure that it's inchy. This is a super pastel. Wow, look what the green screen's doing to it. This is a super pastel, super gravel, inchy lemonback. This is essentially the all gene snake that we got from this. They're not showing up great on there. Let's do banana, super banana, mahogany, and banana black pastel.
So regular banana, black pastel, and super banana.
3 of the 4 types from that banana clutch. We'll show that. It'll make it a little bit clear. So we knew from the parents. Let me pull up my calculator again. That we had that combination was a pastel, lemonback, gravel, and the sire was a pastel, gravel, inchy. Now that means we can potentially get super pastels, super gravels, and inchies and lemonbacks. The one you saw last is this all gene snake. We had a 1 in 64 chance of hitting it, and we hit it. A super gravel, super pastel, inchy lemonback. Now, how do I know that that's what it is? Well, because I can put it right next to the one next to it and see the difference between what is clearly pastel and what must be super pastel. And I can also see the difference between what is clearly 1 copy of the gravel gene and what happens when you have 2 copies of the gravel gene. And then the lemonback and the inchy are a little bit more of a guess, but we're doing that by comparing how it changes those other snakes in the clutch. And so you just compare all 7. If I showed you a picture of all 7 of them, which we'll do a video of all 7 of them later and go, this is this one we think, this is this one we think, this is this one we think. So you're kind of still guessing. Let me go back to full screen. And this who I probably shouldn't mess with because she just ate. So this is a regular banana. You can see, there we go. That's not too bad color-wise. And then this is a super banana. And so by looking at the 2 of them side by side and knowing what potential outcomes we can get, this one in my right hand on the left side of your screen is clearly banana. And the only thing left that could explain why this one is so much lighter is super banana, which we know makes it even lighter. Now, this is a normal banana. The coloration and the pattern is pretty simple and normal. I'll want both of those in a second. I just want to get that out of my hand for a second. This is a super banana mahogany, and the lights are not letting you see the color on this or the pattern, but there is a distinct pattern change that you can see in the photographs that we took with good cameras. I'll trade you this for both of those again. Woot, woot. There we go. Thank you. So this is the same banana that you just saw, and this is a banana black pastel, and it's freaking gorgeous. It is darker. Oh, hey, buddy. It is darker than it appears under these lights right here. And there's considerable pattern. There we go. Now that gives you a good look at both the color and the pattern changes between a regular banana that's in the back and the banana black pastel that's in the front. It looks metallic. Yes, it does. They are gorgeous. Thank you. Yep. My wonderful partner here.
Plop it right down on top.
Boom. All right. Let me get through the rest of the questions real quick, and then we'll call an end to the stream. When someone lists one for several $1,000s, is that a wishful seller, or is it really that rare? Could be either one, WJ. Some of the ones I think are listed incredibly overpriced, but some of them are just very, very rare, especially when you start looking at snakes that have 3 or 4 recessive traits. Like if you had a piebald lavender hypo desert ghost, and actually desert ghost is polygenic, we just found out, that could be insanely expensive because it can produce snakes that are heterozygous for all those. Some of them, like when albino snakes first came out, they were like $15,000 in 1992 when Bob Clark, well, it was probably 93 before any of us could get our hands on them. And that's when I wanted to breed to begin with. The thing is, like this, for example, the super pastel, super gravel, lemon back inchy that I showed you, I value that snake at probably between $1,500 and $2,000, just because every baby it produces will be pastel gravel. And when we go through the superstripe complex and you see things like the freeway combination and the highway combination, highway is yellow belly gravel, freeway is yellow belly asphalt, but there's also pavement and puma and all these others. How difficult it is to work the genes into a snake and get offspring. One thing to remember, a male ball python can be ready to reproduce after 1 year. Probably not the best idea, but it's possible. A female ball python tends to take 2 years, 3 is better. And whereas a male can start breeding when they're around 750 grams, a female can't breed until they get up to 1,500 grams. Ball pythons are also notoriously picky eaters, some of them. And so the process of saying, I want to build a piebald lavender, that's 2 recessive traits, means I need to get 2, one male and one female, that both have are heterozygous for those 2 genes, and then breed them together. And where we have recessive traits, let me redo my calculator here and show you. By the way, lavender, albino, and piebald is called dreamsicle. We have some dreamsicles, and we have some het for dreamsicles. So let me show you. So we have we have visual dreamsicles, but if we only had hets, which means we had 2 normal looking snakes that were both het lavender albino and het piebald, then we would have there's a 66% chance that it would be het for both. And then 19% chance of hitting a piebald that's het lav, or a piebald that's 66% chance of being het lav, a lavender that's 66% chance of being het piebald. 1 in 16 gets us the lavender albino piebald, which is a dreamsicle. It looks like this. Actually, this. So that is a dreamsicle, because when you put hets together, you have a 1 in 16 chance of getting this. Now, if we change that so that instead of being het, we actually have snakes that are visually piebald and visually lavender albino. Now, all of a sudden, you pair that to a double het, you have a 1 in 4 chance of getting a dreamsicle. And then if you say, you know what, dreamsicle is nice, but I want a blackhead, leopard, yellow belly albino, yellow belly dreamsicle. So now you have a dreamsicle, which is 2 recessive traits, and then you have added in 3 other traits, blackhead, leopard, and yellow belly. Those are all incomplete DOMs. And now you get a snake that's closer to $2,500. So in some cases, the price that they list at is, hey, I just think I've got something rare and cool that somebody might want. I'm going to put this price on it and see what I can get. And for high-end breeders, who's mostly who you're going to sell to, a $10,000 snake is probably not that outrageous. I don't have any $10,000 snakes. I have a few $1,000 to $2,000 snakes. And yeah. Let me get through the rest of these questions. How long till we see your offerings on Morph Market, or are you already there? We have you saw those 3 banana babies. We have 4 from that litter. We have 7 from the next litter. There are 4 more litters that will be hatching over the next couple months. You'll see Epic Loot Exotic stuff at reptile shows around Texas starting in October. And you'll probably see our stuff on Morph Market around the same time. In the future, hopefully it'll be cheap to know the genes. Right now, a full shed test for all the tests they've got is like $120. I'm getting ready to send off one to verify our Super Pastel, Super Gravel, Lemonback, Enchi, just so that I have the genetic testing for it. And because having the piece of paper that says this is genetically verified to be Super Pastel, Super Gravel means the price I ask is more likely to be met. Personal opinion on old-fashioned farming traits versus genetic modification and forced phenotype expression, then apply that to people if you're willing to share that subject on this stream. So you don't get to genetically modify or choose to breed people because people are not pets or livestock. They're autonomous individuals whose bodily autonomy should be respected. So I don't get to force you to breed with somebody else. But by the way, I don't get to force those snakes to breed. I just put them together and they breed. I'm not genetically modifying anything. But anyway, people ask for food, you ask for snakes. Yes. Years ago on TAA, you said you could make something that looks like a banana appear in your hand, and now it comes true. Fair. Do the observable traits change at all during their development? Yes. Another great question there from Edie Antel. As a matter of fact, let me go back. Well, you saw the regular banana that I just had in my hand. This is an adult banana. It tends to yellow out and fade a little bit. All of them tend to fade a little bit, although there are some genes like Enhancer and Desert Ghost and other genes that are darkening or improving genes that make, like if you combine the right thing with banana, you can reduce the number of spots on it and make it keep its color a little bit. You saw this. You can compare it to the banana that I had in my hand a couple of minutes ago, but also I can show you a baby banana. Nope, that's a banana pod. I don't know why it's listed as, oh, those are sold. You do availability as for sale. There you go. Banana. Boom. This is a 300 gram banana, so it's in between a newborn and this. There we go. This is this is banana yellow belly, so the yellow belly is going to add a little bit to it, but you can see the difference between this and the more grown-up version. Each potential gene is going to change a little bit in what they're going to display and how much they're going to retain when they get older. Well, it happens so much that it might confirm a type that wasn't as apparent before. Yeah, and that's why we don't take, well, we take pictures, but I mean, we don't post pictures or evaluate their genes until after their first shed, because they look a little bit different when they're first born. They look great and their best probably right after their first shed. There are some that, like, for example, in boa constrictors, there's an IMG gene, which is increasing melanin gene. And those snakes get blacker as they age, in the same way that this banana gets kind of closer to a buttery, lemony look as they age. So yeah, there's some things that look like that too, or that change as they grow up. Maybe Monday we'll be bringing home our first ball. He's a leopard Mojave, and we're tickled. Awesome. Leopard Mojave are great looking. Let me find a leopard Mojave. Go here. Do the max rates is true. So, yep, right there is a leopard Mojave. This one was born in 2022, weighs 180 grams. If you compare that to the normal type, and I'm going to switch the normal picture to something like that. Leopard Mojave. What leopard does is it darkens the darks and does some mild pattern modification. And the Mojave adds this kind of contrasty yellow and black. It's distinct, and I have a hard time describing it. If you guys want to know more about the individual morphs, one of the best things that you can do is come over to morphmarket.com. And if you click on, for example, ball pythons, and you can click on traits right here. And what you've got is a list of the available traits. And then you could say, I just want to see what's up with Cypress. Now, if you click on where it says visual count, that will take you to Cypress snakes that are for sale. And this one will take you to the one super that's for sale. But if you click on this I, that will take you to the Morphpedia. And you can say, aha, I love Cypress, by the way. We have none in our collection. I really want to get Cypress in the collection. I think it's gorgeous, especially this white. You see this a lot in the spider complex, and we'll talk about this more when we get to complexes. But yeah, here's the Cypress incomplete dominant. You can find out it's in the spider complex, and then here is the history of it, who discovered it, and what to look for. The head of the Cypress ball python is a light shade of brown that fades toward the neck. The body is usually dominated by a large alien head that tend to display keyholes rather than the usual eyes, whereas yellows and oranges of a normal ball python are. The Cypress expresses various shades of brown and orange. The description's a little hard to tell, but you can go through lots of examples of what a Cypress looks like. I mean, Cypress is one of my favorites that we don't have in our collection right now. All right. I don't know if I got ripped off, but I have a splotched Nelson's milk snake that I got for $600. I can't believe I spent that much on my first snake, but he has been such a sweetie as far as king snakes go. Well, if you love him, then you didn't get ripped off. I like Nelson's milk snakes. I love albino Nelsons. But there are some really interesting, like if I go to my saved snakes, oh, I bet I took it off. I did. There were a couple of aberrant, blotched king snakes that I really loved and almost bought. But some of these things, you can get an unusual looking snake and you can breed it all day long or breed it for years and never get that particular trait out, which means it's probably polygenic. Just like when you breed, you don't look exactly like your parents. When we bought a banana Inchi, the breeder said the Inchi would reduce the spots. So far, it seems to be true. Yeah, Inchi's one of them. It's a little over a year old and just now developing a few spots. Arden, if you're still here, grab Skeletor. Because I think Skeletor is about the best example that we've got for this. 600 sounds about right for a splotch Nelson's. Yeah. All right, Arden's getting Skeletor. We're going to show that off, and then we're going to stop. If one parent's normal, all the kids will have a normal part, even if it's not dominant. Normal is just the default. It is what the wild type is. If you have, like, there are combinations where you won't produce any snakes that are normal in the litter because other genes are dominant. So let me pull up Husbandry Pro. Bloop, bloop, bloop. There's Skeletor. All right. Skeletor is a 5-gene snake. He is a banana, lesser, fire, spot nose, yellow belly. And it would take a... Wow, he looks so good.
He's getting so big.
He's so big. I have pictures of him when he was... Oh, my gosh, we should weigh him. So let me go back to... full screen. This one came from Wilbanks, who's one of the larger breeders, and I trust them to get this right. But the banana part's obvious. The lesser makes it so that he's... I know that the green screen's messing with this. He does not have any spots that I know of. But banana, lesser, fire are all lightning genes, as is yellow belly. And then the spot nose, if you look at his head, you can see... maybe if the camera doesn't wash it out, a little bit of a pattern that looks like a skull on there. And when we first got him, the spots along, or the areas along his dorsal looked like little bones, which is how he got his Skeletor name. Wow, he looks so good. I bet he's, I don't, what was his last weight?
It's like 800 or something.
We go to activities.
It's about 100 or 200 grams.
Oh, no. His last weight at the beginning of June was 666 grams. He was absolutely satanic on his weight. I guarantee you he's almost 750.
Yeah.
And that makes sense because he is just over a year old. Technically, we could breed him. I don't know if I haven't popped him recently to see if his hemipenes have sperm plugs or not. Hold still, buddy. Sorry, I'm not trying to mess with you too much. I just want to, let's do that and that. Yes, he has sperm plugs.
Oh, boy.
Look at that. He's so good.
We're gonna get you
Blue.
fucking.
But we don't know who to pair him to. And one of the difficulties of that, when you have a snake like that that's got 5 genes, is the offspring. How do you tell which genes are in there? And you have to look for very small things, which might be the amount of blushing coming up the side. It might be the width of the lines on the head, eye color. Actually, for bananas, you can also put a flashlight on top of their head. And, we'll do a video on that at some point. We also, by the way, have 2 different black lights that we use. One's a 390 millimeter, or 390 nanometer, and one's a 365 nanometer, I believe. And because of that, it will take the various... So I'll talk about this in another video. Reptiles have chromatophores that determine the coloration. And you go back to the browser thing. This picture will come up next in the next episode when we go through the yellow belly complex. But you have chromatophores that are basically cells that produce different color pigments. And so you have xanthrophores, which are responsible for your yellows, erythrophores, which is responsible for your reds, iridophores, which are responsible for blue-green or reflective. Then you have melanophores, which are responsible for the brown-black pigmentation. So if you have something that is amelanistic, that means it is not producing melanin or not displaying melanin. If you have something that's hypomelanistic, that means it has reduced melanin. And if you have something that is hypermelanistic, that means it has more melanin. So more melanin is a blacker snake. Less melanin is a more ghosty type appearance. And then amelanistic is your albino, where there's no black. Similarly, you can have anearthristic, which is snakes that don't have the red-orange, or axanthic, which are snakes that don't have the yellow. And so... yeah, we'll We'll do a couple other parts talking about genes and taking questions. But if you missed out on the beginning of this, go back and watch. I give a quick rundown with a couple definitions. Nothing heavy. Nothing's going to be too difficult for anybody to understand. It's just about you got two parents. They're passing on their genes. What do we call stuff? And how do we determine, in some cases, by looking, and in some cases by genetic testing, and in some cases by knowing what the odds are and just comparing the snakes that we get, how to tell what we've got. And since this is our first year producing complex snakes like a super pastel, super gravel lemonback inchy, when we've never even had a lemonback before, we'll do some videos showing the comparisons between the baby snakes that came out in that clutch, so that you can see right along with us what we suspect the genes are. And to build off of that, when you're listing snakes at Morph Market, when you look at what most people have for sale, they will have basically the description of their store, and most of the time they'll guarantee sex. Like, we guarantee that you're going to get a male or female, but they don't necessarily guarantee morphs, because some of them are difficult to tell. And what most good breeders will do is say, for example, if I had hatched Skeletor and I wasn't sure whether or not there was yellow belly in there with the rest of it, but I could see the other ones, then I would have listed him as a banana, lesser fire, spot nose, possible yellow belly. Or I might not have listed the yellow belly. Instead, I'll list the pairings of the parents, and here's what I think's in there. You'll find that as well. Because what somebody will say is, we paired this to this, and we're pretty sure this is just a cypress. And somebody else might look at that picture and go, oh, they're wrong. That's a cypress yellow belly, or that's a cypress Mojave, or, whatever else. And then they can buy it and breed it and prove it out. In any case, thanks everybody for hanging out. I had fun putting together a quick take on snake genetics, because we love doing this stuff. And there will be more, but I think what's going to happen next is we'll probably do a video about the clutch of 7 to show how we got to super pastel, super gravel. And then you'll see more. If you go back and watch our past live streams, you can see us ultrasounding our snakes and talking about follicle size and where they are in the gestational process, how we know when they're going to lay or not lay. You'll also see us cutting clutches of eggs open and identifying those first snakes. So you'll actually see us cutting that group of 7, and you can hear me guessing at what the morphs are there. But we'll do an update on them. Thanks everybody for hanging out. We'll see you next time. Bye-bye.